Okay, so just to start out, I'll give you my conflict disclosures. I actually don't have any commercial conflicts that are related to anything I talk about today, but I am the steward of a fair number of NIH awards, and the one that's most important for what I'm working on today is the top one on the right-hand side. I am the PI of one of the civics vaccine centers. I'm actually a frenemy of Florian Kramer because he runs one of the other vaccine centers, and we are both working in the same space along with Ted Ross, who runs the third center. So mRNA vaccines for influenza, I really appreciate what Norbert just did. He really set the stage for everything I'm going to talk about, and I think you'll see some connections in what I'm about to talk about. So if you cross mRNA and influenza in clinicaltrials.gov, you get 34 listings. 11 of them are actually not evaluating mRNA vaccines. They're related to mRNA COVID vaccines and either standard flu vaccines, or somebody is doing flu vaccination and transcriptomics. But 18 of them are actually influenza vaccine focused. And there's a fair amount of work going on in this space, both in the commercial as well as in the academic arenas. I'm going to just pull two slides from publicly available data that have been put out by both Pfizer and Moderna. So this was data from Pfizer's report last year showing that their quadrivalent modified mRNA vaccine could elicit both CD4 and CD8 T-cell responses, and they thought that these results were significant enough that they moved ahead with a phase 3 trial. This slide shows the recently released data from Moderna showing that their multivalent influenza vaccine was able to elicit both hemagglutinin titers, and then the vaccine that included a neuraminidase antigen, as you can see on the bottom right, was able to elicit neuraminidase-specific antibodies. And again, the thinking is that this platform is really going to be useful moving forward, and as I say, there are a large number of trials going on. But the reality is, if you talk to people, everybody's kind of like, why aren't these data better, actually? Given the results of COVID, you know, people are sort of like, they're not as robust as we might have thought they should be. Why are mRNA vaccines really not blowing traditional influenza vaccines out of the water? And I think there are a number of reasons for that, and that's what I'm going to spend the next little bit of the talk going through. So the reality is we need to talk about flu and why it is fundamentally different than what we saw for SARS-CoV-2. So obviously, mRNA vaccines for SARS-CoV-2 have been a huge success, right? It really highlighted the power of the platform. You got rapid development and deployment, but it leveraged previously existing work on sarbecovirus spike proteins. Without that work, nothing would have really jumped to development right away. And it also leveraged all of the prior work on mRNA vaccines and therapeutics, as has been discussed in earlier talks. And these slides are really unassailable, right? When the Pfizer and Moderna trial data came out, I was blown away as well as everybody else. I mean, I don't think anybody could argue that the protection that was shown in the Pfizer trial was anything less than incredible. And the Moderna trial showed exactly the same thing. And in full confession, I was actually a participant in the Moderna trial. I was in a placebo arm. I'm not mad about it. I got rolled over. It was fine. But, you know, the data were really pretty remarkable that these vaccines could do what we had hoped that they would do. But we were in an unprecedented situation, right? We had a pathogen spread in a naive human population. There was not a significant level of pre-existing immunity to SARS-CoV-2 when those trials were performed. We are no longer in that situation, and we are definitely not in that situation for influenza. With flu, the population has high levels of pre-existing influenza immunity. We know that children basically are 100% flu positive by the age of 6. And so there really is no situation where you're going to do an adult trial in influenza where there isn't pre-existing immunity. All of our trials have to be done in that context. There is no cohort of individuals that are flu naive. In addition, we know that flu is globally diverse, and it's really hard to know where it would go next. I mean, we would love to have predictive algorithms, but we're not quite there yet. And this is true for a number of pathogens. So HIV, as Paolo is going to talk about, influenza, SARS-CoV-2, they're antigenically diverse over very short timescales compared to many of the vaccine antigens that we think of as being incredibly powerful, like measles, right? Measles doesn't evolve the same way that flu or HIV or SARS-CoV-2 does. It does evolve, but it's not evolving at the same rate on the same timescale. And so the evolution of these viruses is really facilitating both individual and population level escape. And that creates a challenge for vaccine development. So here's a slide from 2014 coming out of collaborative work looking at the evolution of flu viruses. And clearly flu is continually on the march. And we have some problems as we think about these data, right? We know over time where flu has gone. But the reality is, knowing that doesn't necessarily help us, right? Soren Kierkegaard has this quote that everybody loves: life can only be understood backwards, but it must be lived forwards. And the truth is, we don't know if you immunize with a current strain and look at retrospective breadth, does that mean anything? Or if you immunize with a historical strain and look at breadth as it evolved, does that mean anything? Because we don't have an alternate Earth where we can run this experiment twice, right? We don't know if flu would have evolved in exactly the same way. So it creates a real problem as we think about designing influenza antigens going forward. In addition, there's a huge problem with flu that we've got more than one antigen to deal with, right? Neuraminidase may not evolve at the same speed, but the diversity of neuraminidase is just as bad as the global diversity of hemagglutinin. And the structure of the virus means these things can recombine in any pattern that you want. Now, not all of those are going to infect humans, but we never are able to tell exactly which ones are going to jump. So the combinatorial problem for flu is huge. And so it really comes down to, one, can we make a universal vaccine? But the first question you have to ask is, what do you mean by universal, right? Do we mean a pan H1 and a pan H3? Do we mean one vaccine that will cover both group 1 and group 2? Do we really want to worry about avian cross coverage? All of the Sturm and Drang that's occurring over H5 right now, right? Or, what has occurred for H7, H9, H10? Or do we want one vaccine to rule them all? And that's a laudable goal, but I'm not sure that it's necessarily going to be achievable in the short term. So what are our tools? I mean, really a vaccine is 3 components when you get right down to it. It's an antigen, a platform, and an adjuvant. Now, in many cases, those roles overlap. For mRNA, the platform and the adjuvant are basically the same thing, right? It's mRNA LNP, as was well pointed out earlier in the day. And that being said, you still have to put the adjuvant, you still have to put the antigen in, right? There was a great quote here from 1990 that the best adjuvant will never correct the choice of the wrong epitope. And I would argue that exactly the same thing is true for your antigen. If you put the wrong antigen into the vaccine, it's never going to work. So we really do have to think about what we're doing with these. You have to get all 3 components right. That's the problem. It's sort of like when trainees ask me about which part of the grant is the least important. I'm like, none of it. Everybody reads every page. Make it all correct. Same thing for vaccines. You have to get everything correct. So the mRNA platform is a tool, but expecting mRNA to solve every problem is foolish. And I think we have to think about exactly what it is we're trying to do as we move forward. And I say that as someone who has embraced the mRNA platform. I'm doing it in flu. I'm also working in the syphilis vaccine space. I love the tool, but it is a tool. So there's 2 key questions from my perspective. And really, number one, does mRNA do better than a protein when exactly the same antigen is being expressed? We have data that sort of skirt around this issue, but we don't have a lot of data where we've done exactly the same thing, the exact same protein, comparing it head-to-head with an mRNA version of exactly the same thing. There's a fair number of trials that have gone on comparing mRNA versions of vaccines to currently licensed influenza vaccines. But those are not exactly the same antigens. You've got differences in those products. So I've been lucky to collaborate with the Vaccine Research Center where they had developed this hemagglutinin stem ferritin nanoparticle They did computational design and actually shrunk, got rid of the HA head, truncated it down to a stem epitope that was really focused on the broadly cross-reactive epitopes that everyone hopes will provide broad protection, and created it as a ferritin nanoparticle. And so we were actually able to collaborate with them, get exactly the same sequence, and that has been put into an mRNA. Now, amazingly enough, the VRC 321 trial just got published 8 days ago, and so the data are out, and you can see that in the background of previously immune individuals, that vaccine does increase antibodies that are directed at the stem. And using the neutralization assay that they had, you can see increases in those neutralization titers. And there is cross-protection. You're seeing protection from this H1 only stem ferritin nanoparticle. You're getting cross-protection to H2 and to H5. And they've done B-cell analyses to look at whether or not the antibodies that are being elicited have evidence of cross-protection. And they've been able to show that depending on the epitope that you're looking at, whether it's that central stem epitope or that anchor epitope, that there is cross-protection. More cross-protection, or more, I should say, more cross-binding and cross-neutralization from that central epitope than the anchor epitope. But there is evidence at a cellular level that these vaccines are doing what we would hope that they would do. So I'm actually happy to report that the Civics vaccine trial 2110 has actually opened this month. The clinicaltrials.gov link needs to be updated because we've actually started enrolling. But we are currently enrolling individuals in a dose escalation study for the mRNA vaccine, and we are partnering with the VRC. We will be doing exactly the same analyses. We're going to ask the question, does the difference in platform make a difference in the kind of response that we're getting? I don't know what the answer will be. I'm excited to see what the results are going to be. I think it's an important question to ask. So the other question that I think is key is, what can mRNA do that's really impractical to do otherwise? And I think this is an area where mRNA does bring a lot of advantages to the influenza vaccine space. So one of the things that we've been able to do is to partner with our colleagues at the University of Pennsylvania, both Drew Weissman's lab as well as Scott Hensley's lab, and they had gone ahead and developed a 20 mRNA HA vaccine. So they decided that instead of trying to create the golden ticket immunogen, let's just throw them all together and see what you get. So they did selection of HAs from each of the 18 strains that are present in influenza A, as well as representatives of the 2B, Yamagata and Victoria lineages, and formulated them as mRNAs, and then put them into animals to ask what happened. And these data were published last year in Science. So when you look at each individual mRNA in a mouse, they elicit strain-specific antibodies. And there is some degree of cross-binding, not surprisingly, but in general, the mRNAs were pretty specific for the strains that were being immunized. Where if you take and do a comparison of those single strains and then look at putting all 20 of them together, you get a response that I think is pretty remarkable. So if you immunize with influenza B, you get B-specific antibodies. If you immunize with PBS, you get nothing. That's good, at least our controls work. If you immunize with H1, you get a strong H1 response and group 1 cross-reactivity. If you immunize with H3, you get a strong H3 response and group 2 cross-reactivity. If you immunize with all 20, you get all 20 coming up. And I should note that the dose here is not 20 times the amount of mRNA. The 20 mRNA vaccine is 1/20th of the dose of the monovalent vaccines. So you're still seeing a good response at the same total mRNA dose. And I think that's a really important point. Longevity appears to be quite good. Granted, it's mice. Mice don't live that long. Longevity, experiments take a long time to do, but you at least appeared to have reasonable retention of all 20 responses. And this is a really interesting result. So Scott went back, or actually it was the graduate student who was doing the work. But they went in and they did absorption experiments to ask, is the protection that you're seeing due only to cross-reactive antibodies coming up, or are you seeing strain-specific responses? So with the, on the left-hand side there, with the H1 vaccine, if you absorb with H1, you get rid of that. You don't see any change to anything else with H3. If you absorb with H3, you get rid of the response. With B, you absorb with H1 or H3, no change. But if you look at the 20 mRNA, if you absorb with H1, it only diminishes the H1 response. If you absorb with H3, you only diminish the H3 response, which suggests that there may be cross-reactivity, but there is still a healthy amount of strain-specific response that you are seeing in these animals. Now, when Scott contacted us, we happened to have through, well, frankly, it was COVID is the reason that this occurred. We had a group of non-human primates that had been infected with influenza. We had actually set this experiment up to do some immunization experiments for flu, and when COVID hit, nothing could move forward, so I had animals sitting around eating bananas for a year. And as you can see, the animals had been infected with the 1977 H1N1 strain, and then a little bit later they were infected with the 1968 H3N2 strain. And then they sat around for a while, and we immunized them with the 20 mRNA vaccine. You can see that following the H1 infection, they had a rise in H1 titer. Following the H3 infection, they had a rise in H3 titer. When they got immunized, both of those titers came up. But what happened in this pre-immune model, getting back to what Norbert was talking about at the end of his presentation? We also got our hands on 3 naive macaques. So pre-immunization, no titers. After one shot, reasonable response after a boost, decent titers across the board. When we immunized the pre-immune animals, yes, they had pre-existing immunity to H1 and H3, but getting a single shot, everything came up. Getting a second shot, everything was up. Meaning that pre-existing immunity, i.e. original antigenic sin, did not seem to inhibit this response. And so we are now doing the molecular workup to ask what is going on at the B cell level in these animals. And we are actually asking also, is there any difference in the distribution of these cells? These animals have been subsequently boosted and necropsied, so we are looking at the lungs, the mediastinal nodes. We're asking what was the distribution of all of these cells. So that work is ongoing. These last 2 slides are unpublished data. So my take-home message would be that mRNA technology is a powerful tool, but tools require skill and knowledge to use effectively. And I think that's the key thing, is just throwing the tool at the problem isn't the answer. Using the tool effectively may be the answer. And so I'd like to thank everybody who's worked on this, a lot of people in my laboratory, and then a lot of collaborators. And with that, I'll stop. Oh, yeah, we can go past all of that.